is part of a lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava.
Previously, we have seen that to genetically engineer any organism, you need a gene construct that contains a promoter, gene of interest, reporter gene, and the terminator. Here we will see some mechanic ways to insert the gene construct in the organism we wish to transform. All these methods have in common that they introduce the gene construct mechanically without using any biological vectors. Gene gun or biolistic insertion is exactly what it sounds. Cells are being shot at. But instead of bullets, the projectiles are microscopic beads of gold or tungsten to which many DNA constructs are attached. The gene gun is quite a massive equipment. The tube on the top is attached to a bottle of helium, a gas that will be sent through it at high pressure. Behind the glass pane is placed the cell culture that we wish to transform, and in the middle compartment goes a membrane loaded with the projectiles. When helium under pressure is released through the tube, it propels the projectiles that will bombard the cells. The speed of the projectiles is high enough to breach the cell membrane, and once inside, the DNA constructs are released from the projectile. A very few fragments of DNA will integrate into the genome and transform the cell. If the integration is successful, the cell will be permanently transformed. Bt cotton, which produces its own insecticide, is a genetically engineered crop with the biolistics method. Electroporation is the use of electricity for the purpose of genetically transforming an organism. Cells and DNA constructs are put in a suspension that conducts well. Then short electric pulses are sent in the suspension. These pulses will fragilize the membrane of the cell, creating pores through which the DNA constructs can enter the cell. The microinjection technique uses a microscopic needle loaded with a suspension of the DNA construct to inject it directly in the cell nucleus. This technique requires a lot of dexterity, as manipulating microscopic tools is a very meticulous task. All these techniques have been used to create different types of experimental plants or cultivated crops, but they're not perfect. Probably their biggest caveat is that most of the time, the genetic transformation is not permanent. The gene expression is transient and reduces over time. This happens because the gene construct is perceived as foreign DNA in the plant cell, and it is rapidly degraded by the plant defense mechanism. The only way for the gene expression to be permanent is if the gene construct is successfully integrated in the plant DNA, which only happens under very rare occasions. For a successful permanent transformation, thousands of cells need to be subject to the process in order to obtain a single successful outcome. Another issue with physical transformation of cells is that the cell wall is too difficult to breach. To solve this problem, the plant cells are stripped of their cell wall, resulting in protoplasts with only a membrane to isolate them from the external environment. Finally, all of these methods are quite invasive for the cell and often result in irreparable damage for the cell. Under the ideal conditions of electroporation, about 50% of the cells are electrocuted before any transformation occurs. Despite all these issues, successful cell transformation occurs fairly frequently for these techniques to be still used for crop genetic engineering. Successfully and permanently transformed cells can then be grown into plants using the plant tissue culture technique.